All right, good morning, everyone. So today it's my pleasure to talk about my previous work on the development of the human embryonic limb. So you may wonder why I want to study the limb, because there are a lot of people who study the gut, the heart, and without the gut or without the heart, you will immediately die, but without the limb, you can still survive. But I have to remind you that it is exactly the limb that helped us as a species to create our distinct civilization. So we really appreciate this structure. And also, the limb has been studied in the model organisms in a lot of studies. Sorry, do you know how to proceed? Sorry about that. Yes, the limb has been studied in a lot of model organisms, such as the chick and the mouse. So there has been a lot of things that we understand about how the limb grow and form. However, there are very few studies done in the human. So for me, I really want to understand where and when each gene is expressed in the human limb, and how do they control the growth and patterning of the limb. So to understand this, my previous lab chose the BCM spatial platform, which I don't think I need to introduce this. And we collected actually a very precious resource of the developing human embryonic limbs from the aborted samples in the London network. So with this, and we sectioned the limbs and loaded them onto the BCM platform to get this bulk, equivalent to bulk RNA-seq data. So in order to understand where each population is located in the limb sample, then we have to match the single-cell RNA-seq, and that is precisely what we did. So using a customized pipeline that I developed, I was able to gain higher resolution than other competing atlases, and I was identifying 55 cell states along the 10 major lineages, such as the skeletal muscle and the bones and cartilage. So I want to show you that by combining the two types of data, single-cell RNA-seq and also the spatial transcriptomics, and I was able to review a lot of very intricate fine structures. And this can help us enhance understanding of the cell atlas. So we know when we are analyzing the single-cell RNA-seq data, and the resolution of clusters is never automated. And so, but with the spatial data, we can have an added layer of information. For example, for here, so there are two clusters that are very highly similar, but when you map them onto the space, then you can see one population is around the knee, and the other population is mainly surrounding the muscles. So they are respectively corresponding to the tenocytes and paramecium. And similarly, from the textbook examples, we have known that the distal mesenchyme is related to the growth of the limb. But based on our data, we can see that it is also heterogeneous, and we are able to separate these three populations into a distinct ones that are having different, slightly distinct locations in the foot place. What you are looking at is an embryonic foot, and there are five fingers here, and one population is actually biased in between the fingers. So I was just wondering what's happening, and maybe this is related to the digit growth. And as we all know, we form the fingers from a sculpturing process rather than a sprouting process. So what I wanted to do is to understand what are the gene programs controlling the digits versus interdigit identities. So by doing an unsupervised clustering to define the two groups of the spots in the digits or outside the digits and performing the differential analysis, we can see a group of genes that are mainly expressed in the interdigital region corresponding to the cell death, or another group of genes having the opposite expression pattern, mainly in the finger, promoting the skeletal development. And so using this non-single cell resolution discovery, we can find the gene programs that can be further validated by the single cell staining experiments, showing these very nicely opposing two programs of genes. So, and indeed, actually, these expression patterns in healthy tissue can be related to the genes that are contributing to different phenotypes or deformities that are congenital in human newborns. So I'll skip this. So I think overall, using the single cell RNA-seq and the spatial, we can know a lot more than just having single cell alone. Moving further on, we also generated the single cell spatial data. So we can see fine structures such as the endothelium in between the fingers. So however, this technology has a small number of genes. So here comes a new challenge. How do we integrate all of these three data sets together? And that is an ongoing work in my new lab. So, but to sum up and for acknowledgements, this project was done in my previous postdoc lab in Sarah Teichmann's lab in collaboration with multiple labs, and really appreciate the contribution by this beautiful country and also the kind of women who donated the fetal materials. And now I started my new lab, and hope you will, if you are interested in the city of San Francisco, the city with the fog, the sunny weather, and the bridge, and a lot of nerds, please contact me, and this is my Twitter. Thank you very much.
Yeah, you have no time for questions. Sorry. Big omics. This one.
Yeah. Yeah. Okay. Thank you everyone for being here. I'm a bioinformatician at EuroBioC, which is a Swiss bioinformatics solution company. So today, very briefly, I will discuss batch effect very briefly, and I will also present this new method we developed to correct batch effect by nearest pair matching. You all know how batch effects are important, and there are lots of demonstration that batch effects are a serious problem because they mask real biological signals. Unfortunately, they reduce reproducibility, and they also induce false positive to some extent. So ideally, we can have a balanced phenotype representation across batches, which will be the ideal design because batch effect will be buffered up. But unfortunately, study design are often imperfect. So especially in the clinics or in big collaborations where data set is generated in one lab, another in another lab. So this is a clear example of a data set with strong batch effect where sample clustered by technical variation other than by biological effect. There has been a lots of development on batch effect correction methods. Many of them work, others work a bit less, but in most cases, they rely on complex assumptions. And in many cases, especially when batch effects are latent or very minimal, or difficult to catch, they fail to adequately address this problem. So in our company, we deal with really lots of data from clients, from academics and the pharma. And on the side, we developed a method which is inspired by principles of the statistical matching theory. I suggest you to read about this. It's very fascinating for biostatisticians. And the method works using phenotype information rather than batch information. And this essentially split into two main components. A distance-based matching using nearest neighbor search, which is essentially a different way of computing nearest neighbor, conventional nearest neighbor search, which is within and across phenotypes, resulting in a matrix where per each sample you have the k nearest samples within and across phenotypes. Then we extract these labels and construct a vector of pairs, and then we correct the pairs using lemma. You can also correct using other methods, but we've seen that lemma works pretty well. So what's the end of this? We have a matrix which is batch corrected. It needs to be condensed to reduce the size and remove the duplicate signal, but is batch corrected. We perform a benchmarking of NP match against existing batch effect correction methods, and we've seen that it performs slightly well than highly used methods. And for this, we use distinct batch effect correction metrics like differential expressed genes, SNR, silhouette score, or correlation with PC. We also performed extensive testing on public data covering very different representations for batch effect. And we've seen that, again, NP match works pretty well. So what we've done is putting on a paper. The paper is on bioRxiv. All the algorithms is fully available with no restriction on our company GitHub. So please take a look to see all the details. And we're now using, and we intend to use also for clinical research, where in many cases there are donor effect or latent batch effect that are not really addressed by conventional methods. So that's it. Thank you for listening.
Thank you. Any questions?
Thank you. I'll take a look at the paper. I see it's available on the ONIX, but do you have an R implementation of it?
An R? Yeah, it's a package. It's on GitHub. There is all the code, and the package you can install as a conventional R package. And
Sorry, I missed that.
no, it will be also available in our platform. So you can click and use that for batch correction. Yeah.
Thanks. I was wondering if you looked into, in your comparison to some of the KNN-based batch correction methods. I mean, just by the brief description, I noticed a similarity with some of the methods like FastMNN or Scanorama in the single cell field. I was wondering if you looked into this.
Okay, this is deserves two answers. The first is a good question. The first is conventional KNN is different than this method, because what we do in this method is per each sample. First of all, the features are centered per condition group. Then per each feature, we calculate, per each sample, we calculate the K nearest samples within and across phenotype classes. So each signal will be kind of duplicated because of this. And then we correct this, as I said. To answer the second question, this method is not intended for single cell yet. It's for bulk RNA-seq. We believe it also works for proteomics. We also believe that we need to modify a portion of the algorithm to account for, sparsity of single cell and, all the things that affect single cell, dropouts, relationship between mean and variance, all of these. So we will probably do that, but it's ongoing work.
Cool, thank you. Thanks. I see. Are you ready? You have 5 minutes.
Give me 1 minute.
Yeah.
Okay. Hi, everyone. I just would like to kind of give you a brief introduction, a brief overview of what we've been doing so far. IC has been out there so far for quite a while, so 6 years or so, but it still keeps expanding. It's a little bit what you can see in the coming release or more. You see that the IC-verse, the universe of IC and packages is expanding, mostly green. Some failures might be due to the BioC file cache, but everything's fine. You see that Najla told that. We had issues. We had many issues that were like kind of leftovers of we didn't have too much time to think about this together. We finally came together, at least 3 or 4 of us, and we went down from 40 plus issues and 3 long open pull requests. And we wanted to implement a couple of features, and we are now it's ultra sensitive. 20 issues. I just use it. 20 issues, and we have now some concrete plan to how to address these new ideas. That's very fine. One of these ideas is something that we tried to realize, and it was a long-standing thing that I wanted to make it happen. So you're familiar with the selection things that you want to do in IC, and you want to identify these cells because you want to annotate it or re-annotate them. So now this is soon to be possible. It's the sample identification center panel, and not so far away dream is that we can give you right away the code that you would use to annotate that. So take that long time annotation standing problems maybe. And we did this in a couple of hours, following like guinea pigs our own materials to use the workshop on extending IC for that. We can use IC for as a portal, and you can use that in different manners. This is an example that we've been using it into, and now we are working on more ways to actually load the many resources that you can include in your IC instance. And the latest development for that is that you can now use calls within R to basically load anything. So if you have a data package where you are displaying many data sets, then you can be able to do that with IC in one go. I use IC, what about you? Or like Florian, how he would put it, is IC been using Cambridge or is IC been using Cambridge after Aaron left Cambridge? Who is using IC, just by raise of hands? Okay, let's improve on that number by next year. So I'm gonna ask you the same question, but if you want to start using IC, we put together quite a massive set of workshop resources. So this is kind of a full day to 2-day set of learning material that you can use for that. We have been reworking on the IC website so that you can basically reach out to all the things IC in one go or from one place. And we have the new, like a refurbished IC book so that you can read about IC and a little bit of the knowledge that you might need to use to develop essential things about IC, like extensions and all. It was like probably an effort to see if we really knew what we wrote 6 years ago. The next step for that, you can reach out to us if you're interested in training or developing or disseminating. So if you want to learn how to use IC, if you want to learn how to teach to use IC to your colleagues, if you have an idea on how to develop a panel, if you have the idea that your dataset might be more impactful if you just publish it instead of just saying, here's the link to the geo and so on and so forth, and you want a link and you want to make a portal in 5 minutes, that's something that you can do. Thank you for your attention. And this is just a demonstration of a funny extension that Kevin did because he was probably too bored and or really keen on showing you that you can really put anything into that. So it's a shout out to Kevin, Charlotte, myself, and Aaron, wherever Aaron is, in San Francisco probably. And I am happy to take any alpha question probably.
Yeah, you have loads of time, so any questions? You still have stickers, so if you want some. Oh no, okay, you just want stickers. That's fine. What do you think was the most important issue you've closed recently?
We had many issues, many tiny issues. It's just like exiting a teenager phase of many things, just like scrubbing off some leftovers. What's your thought?
The heat was too complex. We put a stop button. Everyone was.
Yes. Oh, that's a good one.
How do we clean the code nicely? Because I just brutally just close the window, and apparently it's not good for the session. So now we put a button, and it tells shiny.
So yes, if your HPC people help to spawn up a container within, and then IC was in there, maybe not having that button could have kept the resources stored, and they wouldn't like you so much. So now you can do that. There was a hand there, but.
Hi, excellent talk. I wanted to ask, so 6 years have passed from the version 1 to version 2 now, I guess. So what in these past 6 years, there has been an exponential growth when it comes to computing, like and the paradigm shift when it comes to AI or something like that. So maybe eventually we'll go towards that direction. I mean,
I wish we had the time to actually explore that or to have explored that one year and a half ago. But yeah, I mean, no one forbids you to do some kind of custom panel where you might implement some interface to large language models and help you assisting you in interpretation of whatever sets you select. So
if anything, the place I would put AI is on the voice feature that we have, because I hard code, you can talk to us and maybe you can update by recognizing some voice command, but they're hard coded. If I could put an AI layer on top of that, saying, you know, Alexa, do or something. Yeah.
I see you do that, yeah.
I see what you did there. Yeah, that's where I would put it first. The rest of it's. Yeah.
I mean, there's a biochatter initiative also from the colleagues in Heidelberg, from Julio Saez's group. So maybe that can give directions on that. I don't know. I mean, if you talk, so if you want to know, let's say, what was the biggest jump between version 1 and version 2, I would say the rework of the entire API for making additional panels development feasible or more flexible, because probably the older one was easier, but was not so flexible. The current one is pretty much like, you can do whatever you want. You just have to know how to do it, and the material that we put together for the latest workshop is probably the best place to start.
Yeah, my question was, do you handle missing values?
We normally don't have so many missing values, but
you do.
Like that. Okay, yeah. So
do you impute?
No, we don't. I mean, IC is not for analysis. We try to avoid any kind of live analysis step, because otherwise, interactivity of the use of the app. So we cannot have that too much. But if you do that in advance, I mean, the NA values are correctly interpreted as they should. If you un-NA them, then you can treat them as any of the other values.
Well, but I ask because many tests fail if you have NA. So you either impute before or...
But again, we try not to test.
Okay.
Usually the panels were locked, but no, the panels are hard code, so they do whatever you tell them.
Thank you.
Don't run for 10 minutes, they run for 10 minutes.
Sarah?
That's too hard.
Because then it skips more than one. Okay. Hi, I'm Sarah. I'm coming to you from the Max Planck Institute for Molecular Genetics in Berlin. And during my master's, I developed an R package around the model that was developed in the lab, which is Center. And okay, so let's get into this. First of all, what are enhancers? Why are we interested in them? Enhancers, as my colleague Persia put, are regions of the DNA, quite short, where transcription factors bind. They get into close proximity with promoters, and this activates the transcription of a given gene. And we know that promoters and enhancers are kind of a first layer of gene regulation, and that the configuration of active enhancers is very cell type conditioned and time specific. Figuring out which gene is being activated by which enhancer is hard, because one gene can be regulated by multiple enhancers, also the other way around. An enhancer can be activating multiple genes, and these are quite complex relationships. And at the same time, we know that uncovering gene regulation will lead to a higher understanding of how phenotypes arises in different diseases. And there are also a lot of mutations related to non-coding regions, which we don't really know that much about. So our motivation was to develop an easy-to-use tool that can predict cell type-specific enhancer-target interactions with little input as possible and interpretable features. So as you heard yesterday, CROP has an enhancer targets step, but that is a lot easier. Ours is more general and more fine-tuned, and it came after we developed that and we learned from that. And so, okay, what do we need from the user? Is there a pointer? Oh, shit.
Crop.
Okay, so from the user, we only require ChIP-seq for 3 histомodifications. These are quite routine experiments, and you can find them for many cell types, and also RNA-seq, or for one cell type that you're interested in, and also the genes you're interested in. And then from there, we calculate all possible gene enhancer pairs at 500 kb upstream and downstream of that gene. And from the user input, we compute cell type specific features. We also compute generic features. So here, when I say features, I'm saying it in the machine learning way. And these are, the generic features are genomic distance and statistics and cell type agnostic statistics that we computed from very large data sets, many of them that are publicly available. Also, the cell type specific features are based a lot on crop, which Persia presented. Then we feed all of these on a pre-trained to a pre-trained XGBoost classifier. It's pre-trained, you don't need to train it yourself. And then you just get a label for each of the gene enhancer pairs, which says, is it active, is it not active, and a probability as well. Then we compare this to the state of the art, which is closest to ours, which is Target Finder. And we measure the performance on the Benji data sets. These are a benchmark that was done on many cell types using different annotating enhancer target pairs, using different technologies. As you can see, in green we have Target Finder, in orange we have Center. We perform better in some cases than perform better in others. It's quite comparable, but we're using much less data than they are. And we compute around 30 features on our data, they compute 300. And so we feel our model is much more understandable for people as well. So yeah. And as you can see as well, if anyone noticed, F1 scores, the higher they are, the better. F1 scores are still very low. This is a general issue in this field because we don't really have a gold set standard. The closest that we have is Benji. As you can see, even within the same cell type, the performance varies greatly. This is because the data sets in themselves are also very different. The size in them is also very different. And yes, let's go. Okay, so acknowledgements. I want to thank Professor Martin Vingron, which is the head of my lab, and also Dr. Tristan Rapakulia, who's the person who developed the model. You can find the R package in this QR code. You can also find the article, which was published last year in Bioinformatics, if you're interested in more concrete details. Thank you. Questions, anyone?
You're out of time.
I'm out of time, okay. Ooh.
Thank you. And then, and again, it's going to finish his previous talk.
Hope you don't mind. Okay. Hi, it's me again. So I hope to entertain you for another 5 minutes. So this is actually a sequel to Antonino's talk. We can't stress enough that batch correction or data integration is not a solved problem. And here I just show you some cartoon diagrams. And we see that without correction, then usually the same sample, even for the same sample and measured from different labs, put together, you still see the overall transcriptome dominated by the technical variances. And although there has been a lot of efforts trying to integrate the data sets, we usually suffer from the undercorrection where we have a partial removal of the technical variances, or overcorrection where we actually remove a lot of useful biological signals. So now I want to take a step back and then have a reflection on our previous efforts on the batch effect correction and data integration. So for the single cell world, starting with the MNN, the flagship and the pioneer algorithms to do the mutual nearest neighbor to try to squeeze the counterparts between batches together, there has been a lot of efforts trying to match the similar cells or neighborhoods between different batches, and then use that to integrate different data sets together. However, we notice that usually it has the overcorrection problem. So then an independent group of methods have been developed where they use the biological meaningful labels, or what they call the cell type labels, as another input. And then they try to use that to protect the variances that are associated with the useful signals. However, this method is also not very successful. And we can also naturally think that the cell types or cell classes are not just discrete labels due to either biological continuity or the uncertainty from the sparse single-cell RNA-seq data. And more recently, there has been a new group of methods based on the foundation models, and the GeneFormer and SGPT trains a large foundation model, and then they fine-tune the model to perform the data integration methods. However, there are very few evaluations on how this performs. But overall, this group of data integration methods, they all treat the batch effect as a black box. We never learn anything about the nature of batch effects. And every time we have a new integration method, we retrain the model and create a bespoke model to solve the problem. But have we ever done anything that has inspiration from the assumptions of biology? And have we ever tried to incorporate our prior knowledge to understand and correct the batch effect? So actually, there has been another word that people have maybe sometimes forgotten. So for a lot of algorithms that we use.
1 second, can you speak into the microphone?
Okay, for a lot of methods that we use for data cleaning, actually we use the prior knowledge, such as for some of the algorithms where we infer the cell-free RNA, or we call the ambient RNA, or for some algorithms, we remove the certain neuron gene size to correct for the cell cycle and remove the contribution, or remove the stress like AP1, immediate early gene signatures. So, and also there are methods that use the co-expression to further enhance biological signals. So these are all based on assumptions. And what this reminds us is that actually the so-called batch effect is complex and it's multifaceted. And so different groups of genes contribute to the transcriptome in different ways. So here is just a conceptual diagram for the genes in the whole transcriptome. It can contribute to both the cell type, cell state, cell cycle, or batch effects or a noise. And what we have been doing for highly variable gene selection is just to remove the central part, but we never take care of the other parts. The batch effects can actually be decomposed into multiple aspects of the transcriptome. So this has barely been done. So what I propose is that in future, we really need to understand what batch effect is. So we probably will have to do a lot of large retrospective studies based on the previous well-curated labels of metadata and biological data. And also we want to do what Antonino has been doing, which is to rigorously benchmark different methods to see what is the best to create the well-integrated cell atlas. And the goal is that after doing all of these efforts, we want to create some high-quality integrated reference atlases. And in future, maybe we no longer need the data integration or handle the batch effects. We can just use the reference-based analysis, just like what we do when mapping the RNA-seq reads. So overall, this is just my thoughts for the batch effect, and this has been wrapped up in a perspective under review. And I want to acknowledge my co-writer, Sean Lee, and also these three, Mota and John and Sarah, for giving the input of this perspective. And happy for discussions. And finally, in case I don't see you, so safe travels.
You can talk amongst yourselves for a second whilst I get the closing remarks slides. Kevin, do you want to join me? Reluctantly.
I don't want to hide the slide though.
If you want to intervene at all.
Sure.
So this is the last time that we'll be all together in this room, even though there are a few more sessions left. So left on the schedule is so we have the break after this, then birds of a feather, and then lunch, and then you're welcome to leave. So this is the last time we'll be in this room together. So thank you very much for coming. I hope you had a great time. And so thank you first of all to all our sponsors. They, as I said before, they really make the event work without them. There's many things that we can't do that we did. Thanks to the organizing committee, who I say I was very grateful before the event, and luckily no one has died, and that is thanks to them.
Yeah, thanks to Lena, who actually put a lot of work in. So top right, she was in the group of my PI, and put a lot of work, especially with sponsors, and then eventually got to another postdocs in New York. So didn't get to enjoy the conference that she actually put a lot of time organizing. And Simone, I'm pretty sure half the room, if not more, knows her from all the past conferences she's organized. So big help there. And just to go through the numbers of the conference. So there were 110 in-person attendees, and we had 5 keynote talks, 32 posters, 9 package demos, 17 participating countries. I'm really impressed from those that came all the way from Australia. And then we had 20 speakers, 2 birds of a feather sessions still to come, 5 flash talks, and then 25 scholarships made available by our sponsors. So if you want to continue, if this is your first bioconductor conference and you realize I must get more involved, so you can become an expert user, get practicing with bioconductor, join discussions and help people on the support site. Please contribute content to packages. A pull request is always welcome. Workflows, but also books, new data sets. I know Laurie is always looking for package reviewers. There's a few package reviewers in the audience, so come and talk to us if you want to find more about that. Join one of the working groups. You can follow this link for those. And you can apply to join the technical or community advisory boards. And so could those on the technical and community advisory boards stand up? Give everyone a wave. So if you want to join the technical or community advisory board, please speak to one of those people. And then we have more bioconductor events, so more conferences, developers forum, and I think there's a bioconductor event calendar. So the conference website will stay there. The files and videos and links will all be uploaded. And please, if you have any feedback, the feedback does really help us make future conferences better. So that's really useful for us. So please fill out that survey. And then just a quick plug for BioC Asia. Does anyone BioC Asia want to say anything?
So we will have you guys then. You guys. We have an amazing conference in November, as you can see, BioC Asia, which involves all the Australasia part of the world.
And it will be in joint with Abacus, which is the main Australian conference for computational biology. And we will have virtual representation as well. So do we have
So we made a form that I didn't put on the website yet.
Okay.
Register online. So we'll send it around on the website.
Yeah, so that's the website, and please participate. And it's a very nice conference with another community that is often not here because of the distance. So please be part of it.
Great, thank you. And I see virtual participation is available. What's on this slide? So the next US North America BioC conference is in Cold Spring Harvard Laboratory in New York, joint with Galaxy. Maria, do you want to say anything about that?
Yeah, I'll just quickly mention, so it'll be the US conference next year. It will be here, Cold Spring Harbor, expecting about 300 people in person between the Galaxy and Bioconductor communities. Cold Spring Harbor site is lovely. We'll all be staying on the site. So, there's more details on the Bioconductor blog post there, or you can come talk to me here. And hope to see some of you here there at it.
Do you want to say a little bit about Galaxy? Assume it's not the chocolate.
Okay, no, it's not Galaxy. Who knows what Galaxy is? Galaxy project. Okay, a lot of people know. Okay, good. And if you don't, it's a web-based platform for bioinformatics. There are bioconductor tools that are used behind or through it. So this is a way for the two communities to get together and to see, can they identify ways to collaborate more? So build on collaborations that are currently happening and see, can we cross-pollinate?
Thank you. I don't know what this slide is for. It's for the coming slide after that. I mean, I guess from having snug a peek. So breaking news. I mean, next slide, please. We had EuroBioC has been. Thanks to Robert Costello for having done more work before it's even announced than we've done in the first 6 months of organizing this conference. The rooms are booked. The schedule is almost up. Barcelona, and I think it's a nice way of pointing out, like, the weather is turning more and more British by the minute. So we have planned ahead for next year to bring the spirits up again. And we're always looking out for volunteers, actually, for the organizing committee. You don't need to be based in Barcelona. I think from the committee of this year, it started with Oli and me, and then I roped in 3-4 of my colleagues. But the rest of it, like 2/3 of the committee, were not from Oxford or working in Oxford. So don't be shy. Reach out, Bioconductor, in person here, or over the Bioconductor Slack, saying, "Oh, I'd love to have my first try at help." Not even organizing or helping to organize a conference. Put in the words that you like. It doesn't have to be a huge amount of work. It can be about preparing the forms that people use to submit their talks. It can be about processing the talks and organizing, laying out the schedule. Start with small tasks, whatever you're comfortable with, and before you know it, you'll be organizing it in your own institution. Though I'm looking forward to a bit of rest, and I'm sure Oli is as well. So that's what I can think about for Barcelona. One thing that Oli mentioned at the very start about the afternoon session, and again, a nice way of networking with people. I have unilaterally updated the schedule like 10 minutes ago because people kept asking about the TBD, to be defined, of this afternoon. And we've purposefully left it open like this so that people have a chance to essentially use one of the side rooms. I think we still have access, or even in coffee area, set open their laptops, start a hackathon together, or just like keep chatting, planning their next grant, starting IC version 3. It started at EuroBioC in 2017. So, you know, feel free, discuss, use the available space at the venue to do whatever bioconductor thing you want to do. You can use this room or any of the two smaller rooms. So thank you for those that posted photos. If you have any photos left, please put them in the hash photos channel in Slack. It really helps us kind of remember this this conference. I see lots of smiles in these photos, so I hope you had a great time, and thank you so much for coming. I've lost all sense of time. I believe it's a coffee break. You got a question?
Yeah, just wanted to point out as future host of EuroBioC, and the picture that you saw is not only a picture of Barcelona, but it's a picture of the actual venue where the conference is going to take place. And I'd like to say that I've been in the organizing committee for the last 2 years, and I'm impressed with the work that has been done. And I wish that we keep at least with the standard that you had for this year. And I'd like to ask you to join me in thanking Kevin and Oli for the wonderful organization that we have here.
Thank you. I'm looking for keynote 1 here. Thanks.
Be obsessed.